Hello, my name is Harlen Hobbs, and this is my Bioinformatics IRP project. So what I was trying to do was create a program that was able to translate DNA to protein, do T-coffee analysis, and also a little bit of blossom analysis. So here's the way I essentially approached my project. I created the structure chart, which is going to be kind of hard for many of you to understand unless you've had a little bit of programming. But essentially it lays out the flow of data depending on how I did things. And so the most effective way I can show you this is through a short demo. So we're going to go ahead and on the left hand side is going to be my code, and on the right hand side is my actual program running. So we're going to go ahead and A.out that. You can see a menu of items that we can do. But we're going to go ahead and jump right into translate. And so I have pre-prepared a file that has DNA sequences already on it. And so I'm going to go ahead and put that in. And you can kind of see three different frames. You'll notice that there's a lot of the same letters in here. That's specifically because I wanted to make sure every single possible codon was being coded properly. So I just went down all 64 of them and wrote them in. And that's why you see a lot of duplication. But you'll see this top one is the first frame of reference, second, and then third. Starting with the first DNA nucleotide, second and third respectively. And so you'll notice whenever you hit methionine, it starts coloring it green, which is helpful in identifying what would actually be potentially coded for. X's are stop codons, which would essentially cut them out. And you can see that of the three, this is the longest, and this is the only one that actually has a stop codon and is long. One little issue I had with this particular part was the second reading frame. It essentially has a bug where the last protein sometimes won't be a real protein that is available. For example, glycine, it doesn't matter what the third nucleotide is, it just kind of, it could be A, D, T, or C, or A, T, G, or C, I should say. And so because of that, I have a little bit of a bug where it's actually producing a glycine even though it really should be a question mark, which represents not a complete sequence in order to be able to tell what it is. Otherwise, though, it's able to translate perfectly, and it would be able to translate any sequence thrown at it, with the exception of this middle one occasionally throwing out an extra thing. If we were to look at my T-coffee function, it's able to ask for a number of protein sequences. If I do anything too high, like 7, it'll tell me that I can't do anything except for between 2 and 6, and that's got to do specifically with the programming involved. So for every single level of comparison, like if I'm comparing 2 protein sequences or if I'm comparing 3, I had to code it completely different because you compare 2 things differently than you compare 3 things, for example. And so if we were to look down here, we can kind of get a feel. Here's comparing 2 on the left-hand side, here's comparing 3, here's comparing 4, and as you'll notice, it just gets longer and longer and more complex. And so 6 was a good capping point for me. And so what I'm going to do is I'm going to put in some DNA, pre-inserted DNA sequences that we can compare. And these are actually based off of our protein lab. And so this is our albumin from different animals. And I'll show you real quick the sequences that I'm pulling them from, so you can see that we started with bovine, horse, human, etc. And so these are all exactly the same sequences that you're about to see in my program, and we're going to compare the accuracy of mine to the accuracy of this program. And so going back, actually it's easier this way. One thing you'll notice is one big difference is that mine actually have color-coded proteins. This helps us see different things like yellow is nonpolar, red is negatively charged, blue is positively charged, etc. So it gives us a quick glance. Anyway, if you look down the line, you can kind of get a general feel for whether or not it's very similar or not. But of note, especially to T-coffee, are these right here. And so obviously the star means a complete match, colon, partial match, period, sort of a match. It's basically, a colon is same size, same charge, period is same charge or same size. And so as I went through this, I compared these results to the actual T-Coffee website, and I found that I had approximately a 70% accuracy. The issue with it not being higher had to do with all the different contingencies that you had to essentially anticipate. For example, you'll notice on the left-hand side, if it was an R and an E, then they had a period. Now, that's a very simple thing to do relatively. I had to do it multiple times because, again, comparing different numbers of things changes things. But when you have so many proteins, you have many different possibilities. And so even though this little section right here is relatively short, I had to do it with every single possible comparison. And so it gets rather lengthy. And so with a little bit more time, I would be able to get a higher accuracy. There's no reason I couldn't get 100% accuracy. Another thing of note is, so these right here represent spacing, basically. And so what this T-Coffee has done is it's just had only the letters, and then it said, well, let's throw some random spaces in here to try to make it match up better. And so they would throw a random space here, 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 and here, and it made it fit better. Now that sounds easy, but you have to have an algorithm that does this randomly. There's no set way of doing this with proteins, simply because it's not logic based. It's just kind of randomly there's a space here, randomly there's a space there. And so my program at this moment can't actually do that. I had to insert these spaces manually. And so that would be another thing to try to improve on in this program, would be to try to improve that. Interestingly, the way that they do that is they basically just have a number associated with each matching, and they do it like thousands and thousands of times before they finally figure out which ones match the best. And so moving on to our Blossom substitution matrix, I was hoping to get into a little bit more depth into this, but on account of time, mostly because I got slowed down in T-Coffee, I had to kind of just stick with the standard Blossom substitution matrix. So these are a list of values of potential substitutions. So cytosine being substituted for cytosine has a score of 9, which is very high. It may sound a little counterintuitive, but essentially the idea is that because there's a redundancy, you could have a mutation that replaces one kind of codon of cytosine for another one. And so it's has a much higher probability than these other things, because oftentimes these other mutations would lead to death. Now, when we use these together, it's kind of interesting to note, for example, right here, pure S's and then a lysine, right? And so it says that they're slightly similar. And if we look over here, we compare, so we go to K and we go to S, and we'll notice it's a -1. So it means it's slightly less likely than completely random. But it's interesting to know that this last sequence right here that has the K is actually from a frog, so it would have had much more time to diverge, essentially. And so what I would have liked to have done, if I had a little bit more time, is so this Blossom matrix is the average for all kinds of proteins. Ideally, I would have had it set up so that it would have specific ones. So for example, albumin, it might not necessarily match this perfectly, although it may have some similarities. And so you can actually do a comparison where you line up all your different sequences, and it generates essentially a number related to two things that may be substituting. So let's say we said T and C or something like that. We would go C and T, so it's a -1, but maybe in albumin it's higher or lower based on its structures. And so if we go ahead and pull up our menu again, you can see the different choices. And then if we were done, we just press Q and we exit right out. Now I'm going to focus a little bit more on the code real quick. Again, you'll notice that there's a ton of code here, and that's mostly because protein doesn't really follow logical sense. It just kind of, you kind of have to code around it to make it work. But it ended up being 1,162 lines of code. And essentially, it was just a lot of time time spent on trying to make it effective with, for example, the different numbers of sequences and other things like that. Anyway, I hope you enjoy this presentation, and I will upload my code so that if anyone ever wants to add on it or just browse it, they have that option. Thank you very much for your time, and